Thank you. So welcome from my side. You can see my screen and hear me, right?
Yes.
Thank you. So today I will have a talk about global analysis of protein arginine methylation. I came from Medical University of Glasgow. And arginine methylation is a universal type of post-translational modification, which involve in many cellular processes. So here you can see 3 distinct methylated arginine occur in mammalian cells, including monomethyl arginine, MMA, asymmetric dimethyl arginine, ADMA, and symmetric dimethyl arginine, SDMA. During protein arginine methyl transferase catalysis, 1 or 2 hydrogen will be replaced by methyl group from some, generating such as side product. Here we can see the 2 key type enzymes, type 1 and type 2, both came from MMA, type 1 further from ADMA, and type 2 further from SDMA. And we can find PLAMT1 is most predominant, which is responsible for 80% formation of ADMA. Researchers already found arginine methylation are related with many diseases. For example, PLAMT1 mRNA increased in breast, prostate, lung, colon, liver, and blood cancer. ADMA could be a biomarker for cardiovascular disease. And our group found in neurodegenerative disease like frontotemporal dementia or ALS. We found hypomethylated RNA binding protein, including HNRMP or FUS protein. But now, for the global level of protein arginine methylation in this neurodegenerative disease is unclear. Arginine methylation also play a key role in maintaining DNA. So knockout PLAMT enzyme will trigger the cellular senescence phenotype. So coming up the question, how does methylation act in pathophysiological processes? To improve our understanding, here we based on NMR to develop an MR-based method quantification of protein arginine methylation. Actually now, for the common methods already have been investigated on Western blotting, mass spectra, and amino acids. Based on previous work, we develop our workflow for arginine methylation quantification. These methods can be used for unlimited biological matrices, including serum, urine, tissues, and cells. So first step, we use methanol to precipitate protein and do the hydrolysis to get hydrolyzase, and then we use chloroform to remove lipids. The next step, we use solid-phase extraction to keep basic or hydrophobic amino acids in elution. And then we record proton resonance frequency based on the characteristic peak. Use the signal intensity, we do the integral quantification. And these methods is not only can be used for global protein arginine methylation, the metabolites which extracted by methanol in supernatant also can be used for untargeted or targeted metabolism study, which means in same batch samples, we can get 2 information. One is protein arginine methylation, another one is metabolites information. So first step, we record MR spectra of standards. Here we can see we use the methyl group of each compounds. And in order to improve resonance, we also record the jealous spectra. Here we can see the ADMA methyl group can be well separated from MMA and SDMA. The overlap peak from MMA and SDMA will be resolved when we change buffer to D6-DMSO. The first step, we did recovery experiments. We use lysozymer to mimic protein environments and use E. coli cell as cell-negative control. Here we can see all methylated arginine can be totally recovered from lysozymer and E. coli cell lysase. The first step, we detect arginine methylation in different cell types. We can find quite high proportion methylated arginine from 1 to 3%. And interesting, we can see cancer cells have highest methylated arginine level higher than the immortal cell line Harcard and higher than the primary cell line fibroblast. And here we use the ratio to show, or percentage to show the data, which is easy in our analysis and also can be used for comparison in different biological matrices. And here we can see the MMA and ADMA show similar changes in different cell lines. Compared to ADMA, MMA and ADMA only occur 10% formation of ADMA, which is consistent with previous work. The PLAMT1 is predominant enzyme. As we mentioned, the protein arginine methyltransferase upregulated in many cancers. So the PLAMT inhibitor already entered into the clinical trials, but there is not any data to show how this works for inhibition of arginine methylation. So we did one screen to see the drug effective. So ADOX, which is the general inhibitor, can inhibit all types methylated arginine. And MS023 and GSK715, which is type 1 inhibitor, only inhibit formation of ADMA. And GSK591, which is PLAMT5 inhibitor, only inhibit formation of ADMA. The combination of GSK715 and 591 shows synergistic inhibition of methylated arginine. So all data are consistent with our expecting. So which suggests our protocol will be a valuable biomarker for drug discovery and screen. Besides that, we also did validation in HPLC. The second step, we test the differentiation in mouse intestinal organoids. So here we use different medium to treat it with the organoids, which will form different cell type enriched organoids. Here we can see more differentiated shows decreased methylated arginine, and which is more predominant in ADMA compared to MMA and ADMA. Here, our collaborator, Professor Bergering group, helped us to do the single cell RNA sequencing. We can find, so for example, for the penis cell enriched shows lowest PLAMT enzyme level, which is consistent with our data, which shows lowest methylated arginine level. And then we're wondering if the aging will be changed by arginine methylation. So here we use mice model. We collect different organs and test different age mice, and we found spleen is most affect organs in aging mice. In the aged mice spleen, we found significant decreased methylated arginine, which could be related with the accumulation of senescent cells. Besides that, we also can find significant increased ADMA, MMA and ADMA in old mice brain tissues. Our hypothesis, this could be related with the neurodegenerative disease, which shows hypomethylated RNA binding protein. And then we did the mRNA level, and we did not find they are, which means mRNA level is not well representative for the arginine methylation. The last step, as we mentioned, some as a methyl donor to the metabolites and biopolymers, and methionine, which can use as the substrate for some, and some provide methyl group for methylation reaction. Besides that, some also can contribute spermidine, and polyamine metabolism. The some cycles are closely connected to folate cycle. The 5-methyl-THF also can provide the methyl group to homocysteine to form methionine. We can find the arginine methylation are closely connected to one carbon metabolism. The metabolites will regulate plant enzyme activity. So here we want to see if these metabolites changes can directly trigger arginine methylation change. So first we test methionine, which is the basic amino acids. And here we can see methionine deprivation significantly decreased methylated arginine. And arginine methylation is an energy-consuming event, which require 12 ATP for one methylation reaction. So here we can see the glutamine deprivation also significantly decreased arginine methylation level. Glycine supplement has been reported to mimic methionine deprivation by inhibiting serine glycine conversion. So here we want to see if the glycine supplement have similar results as methionine deprivation in arginine methylation. In contrast, the supplement glycine even slightly increased arginine methylation level. We are not only tested in HeLa cells, we also did some model in yeast cell line. So in total, we can find decreased methionine, ATP, or glutamine, and increased spermidine and polyamine, both inhibit arginine methylation. And then we follow the dynamic of arginine methylation. Here, first step, we use adducts to treated cells to inhibit arginine methylation, and then change medium, which without adducts, to induce the remethylation. Here we can see for the ADMA, the half-time take 12 hours. And then we also follow the demethylation. Here we directly remove methionine to follow the changes of arginine methylation, which take half time for ADMA, 40 hours, and also for ESDMA. And then we don't want to change any metabolites concentration. We use the isotope tracing. We use 13C methionine instead of 12C and follow the decay of 12C and increase peak of 13C. In all setup, we can find arginine methylation is very slow step. And isotope tracing resolves the dynamic of arginine methylation. So take-home message. So we developed the robust quantification of arginine methylation in biological matrices, and we found which is highly abundant in cells, organoids, and tissues. And arginine methylation can be modulated by small molecules and metabolites. Our study provides insights into the phenotype of cancer differentiation and aging of arginine methylation changes. And when we do the demethylation, we already found one intermediate. So we are in the stage for the assignment. This will be our outlook. In the end, I would like to thank model group and all our collaborators, and for sure, and your attention.
Thank you very much, Fangrong. So keep your question for the breakout room session. And so we'll have our next speaker, who is Henry.
Hi, everyone. So recently we've seen some incredible advances in the field of computational protein prediction. And in particular, this means that someday we could envisage an era where we could rationally predict a protein structure with relative ease. So it would be nice if we had an experimental method to experimentally verify those protein structures. And today I'll be talking about that, where we've been measuring through space fluorine couplings in order to identify contacts within proteins. So it's common for proteins to have phenylalanine residues within the hydrophobic core. So we focused our attention on incorporating these two analogs of phenylalanine, which are CF3 tyrosine and CF3 phenylalanine. And using a polyspecific amino acid tRNA synthetase, with a amber stop codon at the incorporation site. We managed to incorporate both of these non-canonical amino acids into the protein PPIB. So PPIB has 3 phenylalanines which are close in space, number 27, 98, and 4. And we incorporated these 2 non-canonical CF3 amino acids at sites 27 and 98. And additionally, we looked at another mutant where we mutated this phenylalanine to alanine. And this was in an attempt to create more space within the hydrophobic core to accommodate the CF3 groups. So in total, we had 4 different mutants, and 2 of them we managed to crystallize and solve the X-ray structures. So the CF3 tyrosine incorporation, we observed 2 chains within the X-ray structure. The first one here had a minimum distance between the fluorines of 3 angstrom, and chain B here had a minimum distance of 5.5 angstrom. Similarly, we also crystallized the CF3 phenylalanine mutant, and this time with the alanine mutation at site 4, which allows the CF3 groups to come a bit closer. And this had a closest fluorine distance of 3.5 angstrom. So by simply taking a one-dimensional fluorine NMR spectrum, we can immediately resolve all of the signals from these mutants. So we did these measurements at 400 megahertz because CF3 having a relatively large CSA meant that the lower field gave more favorable sensitivity and line widths. And we were observing a line width of about 15 hertz here. And you'll notice the excellent chemical shift dispersion of fluorine means that we can always resolve these peaks nicely for all of these different mutants. So all of our mutants also produced through space couplings, which is quite surprising when some of the distances are out to 5 angstrom between the fluorines. So here I'm showing the spectra for the CF3 tyrosine mutant with that F4A mutation. And even in the one-dimensional fluorine spectrum, you can almost resolve the couplings themselves, which are about 5 hertz here. But this was processed with a resolution enhancement window function with a Lorentz-Gauss shape. So a much more obvious way to observe the coupling is through the large cross peaks that we observe in the DQF COSY here. And this is quite a sensitive experiment. And you can see a bit of a roofing effect also in the spectrum due to the close proximity of these spins of the chemical shift. So here I've got the CF3 tyrosine, but without that F4A mutation, and the phenylalanine pushes the 2 CF3 groups a bit further apart, and consequently we get a smaller through space coupling for this sample, and smaller cross peaks within the COSY spectrum as well. But we noticed a rather interesting shape for the diagonal within the COSY spectrum here, where the positive peaks have sort of coalesced to the center. And we managed to explain this arising due to cross-correlation between the dipole-dipole relaxation within the CF3 group and the CSA relaxation. So on the right here, we successfully simulated that same spectrum using the MATLAB software Spinach, which has been developed by Ilya Kuprov. And there I modeled the system with coordinates from the crystal structure and a CSA tensor of 25 ppm. which very nicely recreates the experimental spectra that we observe. So in order to quantitatively measure the through-space coupling, it's actually quite a challenging process for these samples, because the coupling constants that we observe are between 2 and 5 hertz, and yet the line width is around 15 hertz. So it's not a value we can resolve easily. So we devised this experiment, which uses a series of jump return elements to selectively turn on or turn off the homonuclear coupling. So by incrementing this delay tau, we can track the evolution of the coupling and the relaxation within this first experiment A. And then in the next experiment, by using a selective refocusing element with this jump return, we only evolve the relaxation. So then if you take the ratio of these evolution curves, you can remove the relaxation component and just keep the coupling evolution. And by fitting a cosine cubed function to this ratio, we can extract the coupling constants for the through space coupling between fluorines. So you can see here for the CF3 tyrosine, we have a pretty small through space coupling of 2.5 hertz, but with that phenylalanine to alanine mutation in the CF3 groups come a bit closer together, and we get a larger coupling of 6 hertz. So quite remarkable that there's actually enough sensitivity for a peak that broad that we can still extract these coupling constants. So in order to quickly identify these contacts between CF3 groups, we've discovered that the TOXI experiment is actually quite sensitive. So within half an hour, with a relatively dilute protein concentration, you can detect cross peaks within the TOXI spectra. And here I'm just comparing that to the NOSI, where the cross peaks are relatively weak. So it's really quite a convenient experimental setup, because on a low field spectrometer within 30 minutes and no cryoprobe, we can detect that these contacts are available within a protein. And there's no need for chemical shift assignment at all, because we know that we've incorporated only 2 CF3 groups. So hopefully it could be a useful effect to use. So thanks to Haocheng and Eden for preparing the samples throughout this project, as well as Rebecca and Colin, who have solved the crystal structures and crystallized the protein itself. And this project was supervised by Gottfried Odding and Thomas Huber at the Australian National University. Thanks.
Thank you very much, Henry, for the really good talk. The next speaker is Philip.
All right. So, yeah, thanks. So first, I would like to thank the organizers for giving me the opportunity to present our data here. I will talk about the activation of the dead box helicase DBPA by ribosomal RNA of the nascent ribosome. So first, a short introduction to dead box helicases. So dead box helicases are ATP-dependent RNA helicases that can non-possessively unbind short stretches of duplex RNA. So the dead box helicases consists of 2 RAG-A-like domains that tumble independently in the apostate. But when ATP and double-stranded RNA bind, there's a conformational change to a closed conformation. And this leads to a destabilization of the duplex RNA, and then one strand can dissociate, and then this product bound state here is formed, where ATP and single-stranded RNA is bound. And this state has been very well characterized using X-ray crystallography for many different dead box helicases, where it looks basically identical for all of these helicases. And in all of them, the ATP is sandwiched between the two REC-A-like domains, and the RNA, or the single-stranded RNA product, is bound on a bipartite RNA binding site that is formed by both REC-A domains here on top. And the RNA conformation in this state is not compatible with the duplex formation. And ATP hydrolysis is rapid in the state, so the ATP is hydrolyzed, and then the products can, ADP and single-stranded RNA can dissociate, and the cycle can start again. And so the double-stranded RNA bound state is not that well characterized, but based on thread experiments, it looks probably similar to the close conformation bound to single-stranded RNA and ATP. So this close conformation is like the central part of the unwinding process. And I work with the dead box helicase DBPA from E. coli. It contains, in addition to the REC-A core domains of the helicase, a C-terminal RNA recognition motif, which specifically binds to hairpin 92 of the 23S RNA during ribosome biogenesis. So this recruits DBPA to the nascent ribosome. And interestingly, DBPA is not active as a helicase unless the RRM binds to hairpin 92. This then activates the helicase core and allows the unwinding of DBPA to start during ribosome biogenesis. And yeah, the crystal structure of the RRM bound to a 23S RNA fragment has been solved some time ago, and it shows that the RRM basically only recognizes the loop of hairpin 92. And DBPA has also been used in many studies to gain insight into the unwinding mechanism of dead box helicases using artificial substrates like the one shown here, where hairpin 92 is fused to a short duplex whose unwinding is then studied. And so we are interested in this activation mechanism, where we wanted to find out how does binding of hairpin 92 to the RRM activate the helicase core. To this end, we used methyl Trosi NMR. So we deuterated and labeled the methyl groups of DBPA, which gives a quite nice spectra for 50 kilodalton protein. And we assigned the methyl groups. And so from now on, I will only show you the isoleucine region for simplicity, but we also include the data from the other methyl groups, of course. So yeah, to gain insights into this activation mechanism, we started with RNA titrations. So we titrated this RNA shown here, which consists of hairpin 92 and a 5' single-stranded region of 40 nucleotides. This RNA can also be used in unwinding studies when another RNA strand is hybridized to the single-stranded region. So binding of this RNA to DBPA leads to large chemical shift perturbations in the RRM. So the RNA binds to the RRM with the RNA binds to the RRM with a hairpin 92, but does not really contact the core domains. So we don't see any influence here. We can also induce the formation of the closed conformation when we add the non-hydrolyzable ATP analog ADPNP. And here you can see there are really large chemical shift perturbations in both REC-A domains. And we also have NOSI data to show that it's really the closed conformation that is formed here when this RNA and the ATP analog are present. So we have this RNA here with the hairpin 92 and the single-stranded region. And to see if both of these parts are necessary for the formation of the closed state, we titrated either to single-stranded RNA or the isolated hairpin to DBPA, and then added again the ATP analog. And in both cases, we don't see the formation of the closed state, which means that we really need the single-stranded RNA and the hairpin 92 at the same time to get the closed state, which is of course essential for the unwinding process. And this means that probably hairpin 92 has to bind to the RRM, and then single-stranded RNA has to bind to the core, and of course ATP also has to bind to the core to get the closed state. And we can also replicate this when we add single-stranded RNA and hairpin 92 in trans, so not in one construct, and ADPNP, then we also see a partial formation of the closed conformation. And yeah, so somehow binding of hairpin 92 to the RRM favors the formation of the closed state. And to find out how this might be achieved structurally, we solved the structure of a a DBPA construct that contains the C-terminal rec-A domain and the RRM. As we have shown before that there are no contacts when the RNA binds to the rec-A domains, but the only construct that was soluble was, the minimal construct that was soluble was the RRM plus the linker and the rec-AC domain. So this is already an indication that the two domains are somehow connected. And our structure indeed shows that there's a tight connection between the two domains, where the RRM binds here to the alpha helix 3 of the rec-AC domain. So the structure was based on amide proton method NOEs and Talos backbone restraints, because the quality of the structure was not sufficient to look at all the side chains. So we had to deuterate the protein. And you can also see that some of the loops are rather are not very well defined in our structure, and this is due to dynamics, as we can see based on proton 15N head NOE values. So we have this tight interaction between the two domains, and to find out how this might contribute to the formation of the closed state when hairpin 92 is bound to the RRM, we produced a model of the closed conformation of DBPA, where it is bound to the RNA that we used for the titration experiments that contains the hairpin 92 and the single-stranded overhang. So we modeled the hairpin 92 based on the crystal structure that I showed in the beginning. So the interaction between hairpin 92 and the RRM is known. Then we used our structure, which shows the orientation of the RRM and the C-terminal rec-A domain. And then we used the orientation of the two rec-A domains that are known from many crystal structures of dead box helicases in the closed conformation. And then we modeled the single-stranded overhang here in the active site on top of the two rec-A domains. So, and this shows, interestingly, that the RRM positions hairpin 92 in a way that the stem of hairpin 92 interacts here with the part of this N-terminal rec-A domain when the closed conformation is formed. And this patch here is positively charged due to three highly conserved positively charged residues that can then interact in this conformation with the stem of hairpin 92, which of course would explain how the closed conformation is stabilized when hairpin 92 is bound to the RRM. And to validate our model, we used a couple of experiments. So we did PRE measurements where the RNA is spin labeled either at the 5' end or at the 3' end, and then recorded methyl group PREs shown in red here. And you can see the PREs that we expect based on our model are in very good agreement to the ones that we observe in the measurements. Then we also did NOSI spectra for the closed conformation. And here we can observe NOE contacts between the RRM and the N-terminal rec-A domain shown in cyan, and an NOE contact between the RRM and the C-terminal rec-A domain shown in red. So the domain orientation is, or the NOEs also confirm the domain orientation of the RRM relative to the two other domains in our model. We can also show based on immunoproton spectra that the stem of hairpin 92 is still present in the closed conformation. So this is not disturbed when the closed state is formed. You can see here the three immunoprotons from the three guanines in the stem of hairpin 92 in the closed conformation. So now that we have validated our model, we also wanted to show that the positively charged patch that interacts with the stem of hairpin 92 is indeed important for the function, so for the unwinding. To this end, we prepared single point mutants of the three positively charged residues, and each of the mutants is sufficient to abolish unwinding of this construct shown here on the right, whereas the wild type can unwind this duplex here. And the mutants also abolish the formation of the closed conformation when we add the RNA that we used for our titrations in the beginning and the non-hydrolyzable ATP analog. For the wild type, we see the closed conformation, but in the mutants, the closed conformation is strongly diminished or not present at all, which indicates that it's really this interaction between the positively charged patch and the hairpin 92 that stabilizes the closed conformation and thereby allows the helicase to unwind RNA only when hairpin 92 is bound. So then we wanted to see if we can also get some insights into the function of DBPA during ribosome biogenesis, because it was known that it is recruited to hairpin 92. but it was not really clear what it is doing in the context of the maturing ribosome, or whether it's just randomly unwinding structures, or whether there's like a specific target sequence that might be attacked by DBPA. And to this end, we modeled again DBPA in the closed conformation with bound to this fragment here of the 23S RNA that was also used in the crystallization study with the RRM. So when we combine these two, then we can see that the 5' region here on top preceding helix 90 is in very close proximity to the active site, which means that this region of the ribosomal RNA somehow is most likely poised to interact with the active site on DBPA during ribosome biogenesis. And to test this, we did again titrations with two different RNA fragments, one that contains this region 5' to helix 90, and here we observe the formation of the closed conformation when we add ADPNP. In contrast, when this part is omitted and we include the 3' region of helix 90 instead, then no closed state is formed, which shows that DBPA indeed preferentially interacts with this stretch here 5' to helix 90. And this might be, either there might be formation of like premature formation of secondary structures during ribosome biogenesis in this region, which might be resolved by DBPA, or DBPA might protect somehow this region here from prematurely folding, because this region of the 23S RNA, this is, folds in one of the last steps during ribosome biogenesis. So it must be kept in an unfolded state during most of ribosome biogenesis to finally fold then in the last steps, and DBPA might be somehow important for this. Okay, so that's it. I would like to thank the people that were involved into this project and for your attention.
Thank you very much, Philip.